Hello, everyone. Thank you for joining this expert panel together with Agilent. And my name is Gema Fuerte, and I am the European Field Application Scientist at Mission Bio. I will be the moderator for today. We will be talking about therapeutic resistance and single cell analysis. So we are very honored to have two excellent guest speakers today. These are Dr. Michael Andreeff from MD Anderson and Dr. Peter Valk from Erasmus. So I'm going to give you a brief description of their work, and then we'll move to some discussion topics. The idea is that I will just ask you some questions. You can give me your ideas, talk among to each other, and the audience can also send questions via the question and answers box. So we will add some questions from the audience. All right, so let's get started with the introduction. So about Dr. Andreeff. Dr. Andreeff received his medical degree and PhD degrees at the University of Heidelberg in Germany, and additional training and faculty appointments at the Memorial Sloan Kettering Cancer Center in New York, in the departments of pathology and leukemia. Dr. Andreeff has been a pioneer in flow cytometry since 1971, when he established the first flow cytometry lab at the University of Heidelberg. In 1977, he joined the Memorial Sloan Kettering Cancer Center in New York and became the head of the Leukemia, Cell Biology, and Hematopathology Flow Cytometry Laboratory. He is professor of medicine at the University of Texas MD Anderson Cancer Center. Dr. Andreeff's group has made major contributions to the understanding of microenvironment-mediated drug resistance and developed strategies to exploit the underlying mechanisms for the treatment of hematological malignancies. So welcome. Now we have Dr. Peter Valk from Erasmus, Rotterdam. Dr. Peter Valk received his PhD degrees in 1999 at the Department of Hematology at the Erasmus University, Rotterdam, on the identification of novel proto-oncogenes in murine myeloid leukemias using retroviral insertional mutagenesis in mice. He worked as a research fellow sponsored by the Dutch Cancer Society in the Department of Hemato-oncology at the Children's Hospital in Boston, US. In 2000, Peter returned to the Department of Hematology at the Erasmus Medical Center in Rotterdam as a scientific staff member and a head of molecular diagnostics. His current research interests are underlying of the biology and improvement of molecular diagnostics of AML and related hematological malignancies. Welcome, Peter. Thanks for joining us today. So, yeah, as I said, we have some questions. I will just ask you some questions about, we just want to have your expert vision and opinion about therapeutic resistance and different single cell technologies and anything that you want to talk about, really. So who wants to get started? I will just ask you, I'll just start in order. So with the first question. The first question is in hematological malignancies and targeted therapies. So what are some of the exciting areas of current therapies in blood cancer today? Who wants to get that one?
Well, I could start.
Yeah.
So for over, well, many years, we have had more or less the same therapy. And if you talk about AML, I mean, this is, of course, my main interest. And only recently we have some real targeted therapies. So initially we of course had the induction treatment, often 2 cycles of chemotherapy and then followed by consolidation treatment. And nowadays all our AML patients are screened for the presence of mutations in, for instance, IDH1 and 2, so the isocitrate dehydrogenases, and also FLT3, of course. And in fact, inhibitors for FLT3 have now become standard therapy in AML treatment of patients carrying these mutations. So things are changing, and that is important, I think, because the treatment has not really improved that much with respect to the type of treatment. And so these are exciting times, but similar for acute lymphoblastic leukemia. I actually just read a paper in the New England Journal, the last issue, in which they also show that even without chemotherapy, you can have very nice and very good responses in acute lymphoblastic leukemia with targeted therapy as well as antibody therapy. So these are definitely exciting times, also with regard to types of treatment, and especially targeted treatment.
Mm-hmm.
Okay, so maybe I can have a different opinion. I think we had a breakthrough with what's called targeted treatment, BCL2 inhibition with venetoclax, but in combination with demethylating agents, the response rates are now 90% in newly diagnosed patients, elderly patients. 3 or 4 years ago, we didn't know how to treat these patients, and the responses were maybe 15%. And interestingly, we are not targeting a mutation here. Just for the discussion, mutation is not the end of all of leukemias. Again, the most successful new treatment is not mutation specific. And I think treatment-wise, we have to get away from mutations and inhibit downstream targets that are common. So now we come to single-cell sequencing. And by the way, there are many other new, very exciting targeted therapies like menin inhibition is taking off. You will hear about that at ASH. Very exciting data. And the BCL2 inhibition will be complemented by concomitant MCL1 inhibition, which in our hands at least preclinically extends survival threefold. Mm-hmm. We haven't seen this before. But now it gets interesting, because should we treat our patients with highly specific targeted treatments that hit only one mutation, or not, or do we need a combination? So the answer is, of course, we need combination therapies, because what we are seeing now with these highly selective therapies, we are selecting for other clones that we are not targeting. Mm-hmm. That's a unique ability of single-cell sequencing, which I think hopefully will replace next-gen sequencing as a routine diagnostic. So Peter, what's your take on that? Is there a chance that how fast will single-cell sequencing replace next-gen?
Well, it is clear. I mean, so if you talk about one of the greatest challenges in our field is, of course, the tumor heterogeneity. And I don't think that is limited to AML only, but it is maybe for many different hematologic malignancies true. And so with next generation sequencing, a diagnosis, what we generally do is, of course, look at the bulk of the cells. Yeah. And then decide, okay, a certain mutation is present or absent, and that is basically it. So we do not really, at least in the HOVON protocols, really look at size of clones, for instance. So that would be possible, but it is obviously limited, because sometimes it is simply difficult to really put the mutation in the right clone, let's say that way. So I do think that single cell analysis will definitely be a step forward in this, because there you would be able to really determine the complete clonal architecture of the sample at diagnosis, and that may also result in different decision making already at that point. The question was, of course, how fast? Well, I'm not sure. It depends also, I think, on the costs of single sequencing. It's always, in my opinion, a pity that we have to talk about euros or dollars, but that is definitely something that is important also. I mean, if it is so costly already to do a single sample, that limits implementation of such kind of technologies. So what will be important, that we will nevertheless introduce single cell sequencing also already at diagnosis, and show show that it really has an additive value, and then things will probably become cheaper. Like next-gen sequencing has become cheaper over the years already. So I mean, this is going to happen anyways.
So I was saying for that, because let's say we have a minor subclone that is being missed by next-gen sequencing. like P53 or FLIC3, we should target that early on before the clone becomes the dominant clone, right?
Yeah, yeah. Well, and especially in the era of clonal hematopoiesis and hematologic malignancies, it is often challenging to really discriminate one from the other. And it's clear, and that is actually also has been shown also already by single cell sequencing, that you can indeed discriminate between clonal hematopoiesis and the true leukemia. And that may also absolutely be of interest if you look at the patient at diagnosis and look at the clonal architecture to really discriminate between the two. So clonal hematopoiesis and true leukemia. And that obviously will not only be important for the patient at diagnosis, but maybe even more important when a patient is monitored during the course of the disease. And I think that will be interesting also to how can we better distinguish between the two also in a setting of minimal residual disease detection. And there we can hopefully also delineate the constitution of the minimal residual disease that is still present. And that may also, well, certain constitutions may be more associated with the impending relapse than others. And that will definitely be of interest as well.
So on that note, Peter, so besides costs, right, that you're saying that, you know, it might be right now, like it was next generation sequencing before, expensive to do it to everybody. What else besides cost would you like to see for the current single cell technologies that are out? Because it's only because Michael, you were also saying that like only mutations. So perhaps you are also, you would like to have the protein information and the RNA, all of them, whole transcriptome. That's the sort of question I'd like to know.
Right. So what's missing is bioinformatics. We need more robust bioinformatics pipelines. It's incredibly difficult to decipher this data. And so we get wonderful help from Mission Bio. Thank you very much. But at one point, you know, this has to be automated based on the data without too much human interference. Otherwise, you can't get it into a routine lab. And then the next step is just to do surface markers at the same time that we do mutations is not enough. We need a whole spectrum of intercellular signaling molecules, phosphoproteins, proteins that we can target and that are relevant. This will not happen so fast. It will take a few years, but there is a convergence of data, RNA-seq data, and the more proteomics, the better, because RNA data does not translate very well into protein data. We have no idea about splice variants as a consequence of that, phosphoproteins. So my emphasis would be on proteomics, and we are doing this independently by CyTOF, trying to merge CyTOF data with DNA sequencing data. Not easy.
No, certainly. What do you think, Peter? What is your opinion?
No, I fully agree with Michael that this is probably very challenging to do it. I mean, we would like to have the whole package, right, of the cell. But I mean, so I think that Michael's really pioneering here to do the CyTOF analysis. I may be more in the ballpark where I want to keep it a little bit simpler and maybe focus on the mutations and the RNA analysis. And one of the challenges today is actually that, so with Mission Bio, you can do very good genotyping. With the 10x platform, you can do beautiful gene expression profiling. And well, we really would like to connect these in one of the two technologies, or maybe link one to the other. I think this is, I mean, one of the challenges that we face today. And maybe we even need other platforms like nanopore sequencing to do this also. on a single cell level or a combination of these technologies. But this is what we would like to do, obviously, and at some point, as Michael indicated, also on protein level, because RNA is yet again also limited. If we have the gene expression profiles, the protein profiles may be completely different yet again. So that will definitely be challenging. But so Michael, if you would like to get this question, so how long would it take to do protein analysis and also phosphoproteomics in single cells to be applicable in a way similar to the DNA and RNA analysis we do today?
So we are doing that with CyTOF, and the uncertainty there is to identify a leukemia cell. But that problem is even worse with RNA-seq, single-cell RNA-seq. Yeah. We have major problems. We have now developed algorithms where we use aneuploidy as a defining marker, but that's the best way and the safest way to identify leukemic cells. I have a big program on minimal residual disease in AML, focusing on p53 and FLIC3. And it has been a nightmare to identify leukemic cells by RNA-seq. And so we then thought we play it safe. And since we are interested in p53, 80% of complex karyotype patients have this mutation. That means we have aneuploidy, and we use the known aneuploidy to identify leukemic cells. But the step to really high dimensional proteomics will be difficult.
Is it possible to so because we now have the antibodies and the leukemia-associated immunophenotypes and possibly the immunophenotypes of the hematopoietic or leukemic stem cells. So is it possible to combine those with CyTOF or not?
So we are doing this. We have 55 antibody panels for CyTOF now. And one could, let's say we're interested in stem cells, so we would pick cells that have CD34 high, 38 low, 123, CLL1, and we would pick a cluster in CyTOF, and then we pick a cluster in by RNA-seq and merge the data. I'm not a bioinformatician, as you can hear from my language. So we need bioinformatics here big time to unify these data sets. Ideally, we would all do it on the same cell.
Yeah, I agree.
Okay, here I have a question from the audience, if I may. So one of the biggest challenge for gene expression analysis with NGS is batch-to-batch variation and lab-to-lab variation. While multiple different bioinformatic tools have been proposed to normalize the data, is it still challenging to find its application in diagnostics and patient treatment decisions? Do you think single-cell RNA sequencing has future in diagnostic patient stratification?
Well, that is a great question. So I have been involved in a gene expression profiling almost 16 years ago. In those days, we actually did it with on the bulk of the cells, and we looked at gene expression profiles, and we also tried to translate it to assays that would be applicable in the clinic, and we were a little bit successful, but this is a super challenge. So to really standardize it in such a way that you can indeed apply these kind of technologies, especially gene expression profiling in different labs, using different platforms, etc. So if now the question is, okay, how would this work for single cell analysis? So I think it's really difficult unless you would be able to review a certain gene expression profile for a subset of cells that would be one way or the other predictable for outcome. But I wonder if that is really possible. Maybe in an MRD setting, so maybe later on that you would be able to recognize gene expression profiles in residual cells that may be relevant to confer some sort of resistance to these cells. I think it's quite difficult to do such analysis at diagnosis. Seems to be like being very challenging.
So my take on that is that the MRD cell is different from the leukemia initiating cell. And these are the cells that survive or have survived treatment, and these are the cells that kill the patients, not the leukemic stem cell type. That's why we are focusing on MRD. And we are not, I don't see my job as identifying MRD. There are hundreds of papers showing that MRD is more important prognostically than everything else. We're looking for new genes that we can target. So, for instance, in P53 mutant patients, surprisingly, we have about 45% responses to chemotherapy. So there must be a difference between a P53 mutant cell that responds to chemo and one that doesn't. So that's the hypothesis that we are pursuing, that there is a difference, and we are trying to identify this difference. And we are using RNA-seq to do that, single-cell RNA-seq, and of course, Mission Bio.
So perhaps now that you are talking about the techniques, you already you mentioned some techniques that you use. Why don't you say tell us what single-cell approaches are you currently implementing, in case you have any other, new techniques at the single-cell level?
Is this me or is this Michael?
Peter, you can start. Yeah.
Yeah. So currently we are looking at minimal residual disease, and we did this in the bulk of the cells previously. And so I do think that it is very important to know the order of mutations that occur in these cells, and there you may be able to discriminate between, or you will be able to discriminate between early and late mutations. And residual early mutations without having the late mutation, that's actually what we showed, that there's not such a strong association with relapse rate as compared to the late mutations. But some mutation may, when they occur early, be an early mutation and not associated with relapse, whereas the same mutation could occur later and then may indeed be associated with relapse. So in that sense, it will be important to know the order of mutations at diagnosis, but also, my light goes off, but also the in the MRD sample, as I just said. So, okay, which cells are now present in the residual sample, and are those still associated with relapse or not? So this is typically, this is one experiment that we are planning to do on many AMLs, because again, I think we can do this on one or a few AMLs, but that will probably not tell us much. So again, heterogeneity, you name it. And then the other experiment that we think of is really to look at pre-leukemia and leukemia, and that is again defined by the occurrence of early only or early and late mutations, and then look at the gene expression profiles. And I think that those are actually quite elegant experiments, because you could do this if you have the right samples of in the same patient. So I mean, this is a big difference to what we did before, where you basically compare one to the other patient, but now within one patient you can look at populations of cells and then determine the profiles of the pre-leukemia and the leukemia and compare those. And that will, I expect, give us important insights into the step from pre-leukemia to the actual leukemia.
So to expand on that, the early mutations seem to be epigenetic mutations.
Yep.
And now, when you have a chip in a 60-year-old individual, only 1% or so develop a heme malignancy. But these patients would be worthwhile following. And once they have an additional mutation, we could think about targeting that. So before they develop, an MDS and then an AML. And we have epigenetic therapies, of course. They are successful to a degree, and they will be more targeted, and more therapies are coming along. So that would be a fantastic application of this technology to identify early mutations and prevent leukemias, right?
Mm-hmm.
We're doing it everywhere else, colon cancer, breast cancer, there's routine screening. Now we have these chip mutations, and if they don't cause leukemia, they're still worth paying attention to because they're involved in cardiovascular disease.
Absolutely. So on that note, I have another question from the audience. And
Michael, a question about this. So what his thoughts are because this would mean that you would start screen older people, right? And then maybe already start treatment with these older people that are not patients yet. And so I'm always a little bit, so I wonder actually, because some people say, yeah, we should do this. Others say, well, okay, if you start doing this, you may in fact already introduce some sort of resistance because of this. And then in the end, you may be not so good off doing this. So I was wondering, what are your thoughts about that? And could single cell analysis give clues there or not?
It has to be at the single cell level, of course. Yeah, so I had a patient who came from another state here, and she had a single TET2 mutation and nothing else, and had completely normal marrow, normal counts. And I was itching to treat her. And of course, you can't treat a patient who doesn't have a diagnosis. And sure thing, a year and a half later, she came back with MDS. And so the frequency is so low, you cannot treat everybody. So you cannot treat a chip alone. That is unethical and maybe dangerous, as you point out. But once we have a second hit, something is happening with these cells, and maybe at that level we could interfere. And the second hit could be a FLT3 mutation, for instance. It takes years for AML to grow out, right? It takes between 1 and 8 years from the first known hit. And so we have time. So some high-risk patients we should monitor. Takahashi in our group published this fantastic paper, in my opinion, where patients had breast cancer and developed MDS later. And of course, they got chemotherapy. Only patients who had a chip, so we had 20-year-old samples, only patients who had chip at the time of diagnosis before any treatment, these patients developed MDS, AML. So that means already practical application. If I'm a breast doctor, which I'm not, and I have a patient with a chip, I will avoid cytotoxic chemotherapy.
Yeah, absolutely. Super interesting. You have already answered the question I was going to ask from the audience, because they wanted to know whether single cell will help you not only at the time of diagnosis or understanding the order of the mutations, but also perhaps making decisions about treatment switching. Because we're talking about, determine what cell started it, what is the order of the mutations, what is the leukemic cell, but whether you could also use it to make a change in the treatment.
Yeah, so let me give you an example, if I may. was published by my colleague Marina Connor-Pepper recently. So we have a lot of experience with BCL2 inhibition. And it took us some time to figure out that IDH mutant patients are very sensitive. And even in the first clinical trial, we started to load the trial with IDH mutant patients. So we did single cell analysis with help from Mission Bio. And the particular patient had 5 clones. One was the dominant clone with a IDH mutation, and then IDH plus NRAS, IDH plus KRAS, IDH plus NRAS again, but a different one, and clone 5 was 0.1% IDH plus another KRAS. Okay, that was before treatment. The blast count was 50%. After treatment, the blast count was 50%. So what do you conclude? It didn't work, right? Wrong. Wrong conclusion. So now the clonal composition had changed, and the IDH clone was vastly diminished, and our 0.1% clone was now 20%. So you can also do 100 patients and figure out that the IDH mutant patients have a better response rate, or you use each clone in a patient as an individual, draw early conclusions, and direct your trials in this way. So that's a good example how we can monitor a treatment and how we can design much better therapies much faster.
Super interesting. Peter, what is your idea? Do you have any 2 cents to add to that?
No, I don't think I have much to add. I mean, so we obviously we also need the therapeutics at some point. So sometimes I feel like that we can do a lot. We can even we can monitor patients nicely. We can do this in an extremely high detail. But then we also need the proper therapies, and we have some, of course. But then you should also be able to translate the results you get from these monitoring and decide, okay, now this is the point to start another treatment. I mean, that so you at some point you're working in such detail that becomes quite challenging as well.
Indeed. So I have more questions from the audience. So one is, what is the plan to address the detection of clonal hematopoiesis mutations of unknown significance in single cells in regards to determining which are a normal function of aging and which ones are potentially indicative of pathology and progression to hematological malignancy? Who wants to take that?
So we started to discuss that. We can't monitor everybody because, but some high-risk patients, and I don't know how to define that. Before I said we should look for occurrence of new mutations, but may also not be sufficient. We may have to look at proteomics and changes in signaling in these cells. If they undergo a major shift in what drives signaling, and it doesn't have to be a mutation, it could be certainly epigenetic factors. So we should monitor the beginning a small group of patients very carefully with all available technologies and try to understand what the next steps are. It may be additional mutations, that's certainly part of it, but there may be other factors, in the level of metabolomics and proteomics, where the cells change their signaling pathways with or without mutations.
Okay, very good. So let me just see what else, because we have a lot of questions. So let me see. So what is the biggest challenge in applying single-cell DNA sequencing-based tests in AML diagnostics or monitoring? Is it sensitivity, throughput, or costs?
Bioinformatics.
But you are talking more about combined, no? Multi-omics or DNA only you would require, you think that you would require?
Me, Yes. Rather standardized pipeline. So, but Peter is much closer to that.
Yeah, well, it is obviously a combination of things. I mean, and definitely including bioinformatics. But I think that maybe even the biggest challenge is the heterogeneity. I mean, heterogeneity in this disease. So we talk AML now, right? Is one of the biggest challenges, because you could go in depth with because you do not have the money to do it, you do not have the patients maybe, and you could go in depth in a subset of patients. But then the question is, how does this translate to all the other patients? And we don't even say, well, that all these patients get the exact same therapy. So that it's not only a heterogeneity in the disease, but also heterogeneity in the way of treatment. Although one would say, well, okay, everybody's treated similar, but it's of course also not the case. So one of the biggest challenges, I think, is the heterogeneity and consequently the number of patients that you need to analyze to get the complete picture. Unless you are interested in a subset of cases, like Michael is interested very much in the TP53 AMLs, then you could, of course, try to focus on this subset of AMLs, which is maybe even another disease, you could argue.
Let me make a more positive statement. I mean, we are curing a substantial number of AMLs, and the cure rates have increased. And suddenly occurs to me, you know, everybody is using polychemotherapy, nobody is using monotherapy. And maybe when we should go back at one day and see in the we can't go back, 7+3 setting, we only have 2 drugs, but clearly they have complementary activity. That means one of the drugs must be more effective in one kind of clones and the other one in the other clones. Then of course they have synergism where all the rest is eliminated in the cured patients. So the answer is we need polytherapy, targeting multiple clones, and we can't throw everything at everybody. Side effects are prohibitive, costs are. So single cell sequencing will certainly guide us for the next set of combination therapy. We are doing this sequentially, for instance, in patients we are treating with crizotinib. We are doing single cell sequencing sequentially, small number of patients, and cytoph to figure out what's the next step, what's the best next combination therapy for these patients.
So hopefully, Michael, you did not get me wrong. So it's not that I'm so negative. So we were talking about the challenges, and I fully agree with you that single cell analysis definitely gives more detail, more insight in what's actually really going on. So it's absolutely very helpful to get more insight into the disease itself and the evolution during treatment, etc. So I'm absolutely positive about the technology.
Think about flow cytometry. When that's single cell proteomics, we just don't think in these terms. But you want to determine CD34 expression in AML bulk by ELISA? I don't think so, because nothing will come out of it, right? So we are doing this already. Single cell analysis, we are basing treatments on it all the time based on flow cytometry, single cell proteomics. Now we are going next level down, 2 levels down. So I'm sure it will be part of it very soon. So it's sequencing has to be lower, costs have to be lower. Mission Bio has to lower the price. Thank you. And the market will explode. And you have to provide a solid bioinformatics pipeline. And with that, I think you should replace next-gen sequencing. Oh, and you have to expand your panel, of course. So we have 100.
You mean different indications, so to larger panel?
No, mutation panel first.
Larger mutation.
50 mutations, and we're covering only top.
But we do custom, so we can customize. The thing is that we have a limit of 1,000 amplicons right now.
Good.
Okay, perfect. So let me ask you another question, which I get asked all the time when I talk about single cell in different conferences. People who are used to do bulk sequencing, they often ask me this question. So if I may, I'd like to ask you so to get your answer. The question is, co-occurrence of two mutations in the same single cell is one feature that single cell and Mission Bio allows you to determine. What is the value if knowing that two mutations are coming from the same cell versus different cells? Who wants to take that? As I said, I get that asked all the time when, when you compare deep bulk sequencing versus true single cell.
Yeah, so I mean, yeah, I'm not entirely sure if I look at the question, but I mean, it's clear that if mutations are present in a single cell and in groups of single cells having the same combination of mutation, it indicates that these two mutations most probably cooperate in the development of the disease. Whereas if you have two mutually exclusive mutations, so that they are always in a single cell solely present, then you talk about different clones, and then they may have the same function or maybe even other function, but at least they do not seem to cooperate. Although you can even think about that they still can cooperate, but I mean, let's keep it a little bit simple. So that would be my first thought. But I'm not entirely sure if I really got the question correctly.
So
yeah,
maybe I can rephrase it. So you have an IDH mutation isolate, and you have a RAS mutation isolation. What's the biological consequence? Then you have a cell that has both. So in the Ross Levine paper that was just published, the IDH mutations have increased CD34 expression, but IDH plus RAS has increased 11B. That's something I would not predict, right? Because you have a very vicious mutation RAS, and the cells are more differentiated. So again, rewrite, and that is just the surface. So there must be metabolomic changes in these cells, which open therapeutic vulnerabilities that you don't have in an IDH only and in a RAS only. So I think that's the answer, that the cells get changed by co-mutations. 1 + 1 is not 2. It's more, and it's different. And so we have to understand what these co-mutations do at the proteomics level.
Certainly, yeah, that's exactly it. I mean, detecting with bulk that IDH and the KRAS mutation will never be, will never give you that heterogeneity and the idea of the different populations, right? That was the idea.
So Gemma, is that always your answer? So you said, I often get this question.
I always get that question because, well, I mean, when you do bulk, it is a population-based analysis. There is no way, even if you sequence very deep, you will find that KRAS perhaps in 0.1 of that example that Michael was talking before, right? Probably. But you miss, you would miss that heterogeneity and what that prognosis of that patient will be in the future by only looking at it with bulk. So that's what I try to answer, the value of having the information at the single cell. And now that we have the protein on top, I mean, of course, that adds much more value, knowing what cell populations are involved. So certainly.
I have another example, FLIX3 mutations. My colleague Koichi Takahashi published that you have the ITD and you have the TKD in different cells. I never thought of that. I always assumed that the IDH comes first in most cases, and then they acquire a resistant mutation, TKD. Not true. The TKD is in a separate cell. It's not the consequence of a pre-existing IDH. What that means therapeutically, I don't know. But the cells are probably easier to manage if they have one mutation each instead of having both mutations.
Mm-hmm. Exactly. So I have another question here. How to effectively integrate RNA sequencing and proteomics or DNA data? How to generate multi-omics data from the same single cell? Peter.
Well, so the short answer is bioinformatics, but I'm also convinced that bioinformatics is not always the solution. I mean, these guys generally also need the data that they can do their trick, to say it this way. But yeah, so there are different ways, and I'm not sure, because we are not at that point yet, what would be the best way. So as I said, so genotyping is very well done with Mission Bio, the expression profiling with the 10x. So how to connect them? So the easy way would, of course, be that one of these technologies would have both RNA, DNA, and maybe at some point even proteomics, etc. But in one machine, because that would make life easier. If you would like to do it today, you may have help with the antibodies and the barcodes, such that you can connect certain population from one platform to the other platform. So that would be a possibility. And then so the genotyping is of course difficult with the 10x platform, because mutations, it depends on or it's the cause because of the way you make your library with the 10x, that mutations that are far from the poly-A tail are simply not captured in this library. And there, what we are thinking of now is to incorporate nanopore sequencing to see if we can amplify the longer reads in these libraries, and then together with the barcode of the single cell, and then see if we can genotype them in such a way that we can have genotyping data and gene expression profiling data as well. How to connect the protein is probably a good question for Michael, I guess.
Mm-hmm.
So what I see is bioinformatics in isolation, of course, doesn't do it. You need give and take. We have meetings, of course, all the time, and they still need us. That we come in with biology, we know a little bit about leukemia, stem cells, biology, and signaling, because it's very easy to come to the wrong conclusions and run array in the wrong direction with bioinformatics. But it will happen. I mean, this is one of the big challenges in cell biology now, and it's a matter of time. I'm pretty positive. It's a matter of having the right teams. You need teams that work closely together. It will happen. A few years from now, we will have it. And we don't understand why it was so difficult in retrospect. You always take things for granted afterwards.
Yeah. So, but Michael, so a question, maybe I'll rephrase a question that I already asked you earlier. So DNA and RNA analysis are now basically done at many laboratories, right? So protein analysis at the single cell level is I think, another challenge. So to really do it on such a big scale as DNA and RNA analysis single cell are currently done. So is that also so you are of course an expert in this, but so do you think that there will be assays at some point that you could do this also at a similar level as DNA and RNA analysis?
So we are at 55 parameters, and we have different panels. We have a stem cell panel, we have immune panels, and maybe we can link these panels. So if you're really ambitious, you do 5 panels, you have 250 proteins, phosphoproteins, and again, if you have luck with your bioinformatics, you could link these panels and maybe link it then to sequencing data. I don't see how you can get this kind of quality at this point from single cell sequencing. by tagging antibodies.
Mhm.
Right now, surface is nice, but it's not not so important. It's a starting point. It's not the end of it.
Oh.
Mhm. Very good. So I see, I mean, we're running out of time. I don't know if anybody else from the audience would like to ask a question. In the meantime, I also wanted, like, more informally to ask you, in these strange times, right, that we are now all working from home and with the COVID, so how that has impacted your research, your work, the clinic, if you want to give us, your ideas on that. Completely moving on.
Yeah, what we have seen in let's say the first wave is that we have seen a drop, and not only on the clinical side, but also at the research side. At some point, the complete research tower here at the Erasmus Medical Center was closed, and then slowly we were able to reactivate research, and the number of patients, non-COVID patients, but acute patients, and also chronic patients increased again, back to normal levels. And well, that is still more or less the same. So with the second wave in Holland, well, hopefully, but it seems that it is sort of under control. I mean, it's very, there are still many patients in the hospitals. But so I'm not a COVID lab, of course. So we do not really feel that yet in numbers of patients that we see, etc. It is now the biggest challenge is not to get your own personnel infected, such that you can close your lab. So that is one of the biggest challenges these days, actually.
So at MD Anderson, we closed down the labs with a few hours' notice. Yeah. Eliminated hundreds of very unique mice, transgenic mice we could keep, but everything stopped for 3 months completely, and then we had a phased opening, and now we are back to normal. The infection rate in lab personnel is essentially zero.
Oh, really?
So we have 22,000 employees at MD Anderson, and we can count with 3 fingers or 1 finger cases in the research labs. So I think MD Anderson Research Labs is the safest place in America.
Yes.
We also have a complete separation between the clinic and the research. So we build walls, physical walls, so people cannot walk across, and samples get through couriers or pass-throughs. Clinically, it's a disaster. I mean, it was a disaster because the patient number dropped like crazy. What happened to all these patients? Nobody talked about the patients who didn't make it to Erasmus or to MD Anderson. Nothing good for sure. In addition, many patients don't do their preventive maintenance, their mammograms, colonoscopies. I got an invitation from my doctor to come for follow-up. Well, I think about it twice before I do it. So it was a big setback, and in theory, we could have all started reading, right? Now we had time to read, but we don't, because I don't know, we're spending so much time having this kind of conferences, which in part are very good. So I do my journal club this way, and it's fantastic. But it wears off. We need, research is teamwork, and it's a water cooler thing. You meet and suddenly something clicks, and it never happens in this format here.
No.
Right now we're in a limbo. We have over 120,000 new cases a day in the US. Over 1,000 patients die every day in the US. And we're all hoping for a spring break with the vaccines, hopefully. But in the meantime, so our place is doing very well. Like the Anderson is doing very well in terms of infections. But very few patients, they get isolated. There is no crossover between teams. So when we have a leukemia patient, we tell the COVID team how to treat them. Not optimal, but we are not going there. So we have safeguards in place and it works out. Difficult times.
Difficult time. You are certainly making it work. So we just need to, hold on a little bit longer until good news about a vaccine came out today. But, this is science, so we need to wait.
Yeah.
Certainly. Okay, so I think we are close to the top of the hour. So I'd like to thank you so much for your time and the time you have dedicated to answer my questions and the audience questions. So I hope to continue working with you, of course.
Thank you very much. It was a pleasure. I think we had a very good discussion here.
I agree.
Emma. And I'm also looking forward to work together with Mission Bio on the single cell analysis.
Yes, very soon. Thank you so much. Have a good day and great evening.
Thank you.
Bye, everyone.
Bye-bye.